Now, I'm so pleased to introduce tonight's speakers. Shahir S. Rizk is an associate professor of biochemistry at Indiana University South Bend and the Indiana University School of Medicine. An award-winning teacher and scholar, he is also an illustrator and poet whose work has appeared in multiple journals. Maggie M. Fink is an adjunct professor of biology and chemistry at Indiana University South Bend, as well as an artist and poet whose work has been featured in exhibits at the University of Notre Dame. Together, they host the podcast Rust Belt Science. Their new book, The Color of North, is an awe-inspiring journey into the world of proteins, weaving together themes of memory, migration, and family with cutting-edge research. The Color of North uncovers the myriad ways that proteins shape all of Earth's organisms and illuminates their remarkable potential to heal our bodies and our planet. Joining them tonight is Pamela Silver, the Adams Professor of Biochemistry and Systems Biology at Harvard Medical School and a founding member of the Wyss Institute for Biologically Inspired Engineering at Harvard University, where she runs the Sustainability Futures Initiative. Now, please join me in welcoming Shahir S. Rizk, Maggie M. Fink, and Pamela Silver.
Thank you, everyone, for being here this evening. My microphone on. Everyone here? Great. Thank you guys for being here. This is my first time in Boston, so I'm incredibly grateful for the opportunity to be here. And my students at IU South Bend are very grateful for a day off of class as well. So Shahir and I are very excited to talk to you about our book that we wrote that was published with Harvard University Press. And there's many things we could have talked about tonight about proteins and why we love them. But I really want to start off with why I fell in love with proteins, and that's this idea of shapes. So everywhere we look in nature, we see this shape show up. Staircase, a spiral, a helix, right? This beautiful iguana tail. Here we see a beehive with that spiral shape. Again, this is something I see a lot of times in my garden with my cucumber trellises. They spiral around. And then again here, this cross section of a seashell. So this spiral shape shows up quite a bit. And if we zoom in to things that are invisible, we see this spiral shape continue to show up. Here is the model of DNA, probably one of the most iconic spirals that we see in nature, right? This double-stranded helix. And this structure was solved in about the 40s or 50s, right, by the Watson and Crick. And this is pretty familiar to a lot of us. We have this idea of what DNA is, what it does. It's the genetic information that's inside of all of us, inside every living thing, is this spiral helix with DNA. And why we wanted to write this book is because a lot of people are familiar with DNA and what genes are, but a lot of people don't know what that genetic information actually makes. It's just instruction manual for something else, and that is proteins. So proteins make up everything that happens inside of our body. Yes, our DNA can dictate what our hair looks like, what color our eyes are, but all of that is because of proteins. Proteins are responsible as well for digesting our food, so that information for how to make those proteins is in our DNA. But proteins don't look like DNA. However, there is a part of proteins that does look very similar. In proteins, we have something called an alpha helix. And this was solved around the same time as DNA was being studied by this man here, one of our favorite chemists, Linus Pauling. And we write about this in the book. He was in bed sick one day, and he was playing around with some of these models, thinking about the chemical interactions that we know exist in proteins and how that could take shape. Just like they were trying to think about what does the structure of DNA look like, he was trying to figure out what was the structure of proteins. And so here's some of his doodles here, and you can see there's this repeating pattern, right? We call this the backbone. And in DNA, we have 4 nucleotides, basically, 4 different chemicals that make up those instructions. Proteins are made up of 20 different amino acids. So the combination that you can have with those is astronomical. So each of these is an individual amino acid that forms this backbone, and decorated on the edges are different chemicals. So they all look a little bit different, and they make up these proteins. And Linus Pauling figured out that proteins can take on this shape here, the alpha helix. And you can see over here as well, this kind of drawn out, this helix structure, but it's very complicated. Everything is drawn out. There's a lot of detail there. And it's informative, but there's a better way to represent proteins. And this idea was come up with by Jane Richardson, who you can see here. She was at Duke University, and she decided that a good way to actually visualize proteins was these ribbon structures, which you can see here. This is an illustration that Shahir drew, where we kind of remove a lot of that chemical detail, and we're left with that backbone structure, that alpha helix. So again, here what we have with Jane Richardson, maybe a little dark, but all these details of the chemistry going on in a protein, we can strip that away and come up with these ribbon diagrams that give us a really nice representation of all the different shapes that proteins can take. And here is an example of one of those proteins. Has anybody heard of keratin? Right? You might put it in your hair, you might take keratin. It's in our hair, it's in our nails, right? It's really strong, it's really tough. And we also see it in the scales of animals like this pangolin here. Okay, and you can see it has these really long alpha helix structures. And this structure makes it very sturdy, very strong. They bundle together very tightly. This beautiful spiraling staircase is an example of one way a protein can look. Here's another example of a protein that has a lot of these alpha helices. They're not long and spindly like the keratin. They're bundled together. This is a protein called spidroin, and this is what spider silk is made of. Again, another really strong substance that we find in nature. So the spider spins this silk out of its body, and it's made up of individual types of proteins, and one of them is this. This is just a small section of one of these proteins, and they form these long strands. And then the spider can actually eat that silk, break it back down into those amino acids, and repurpose it all over again and make more of this silk. But there's also another shape that proteins can take that Linus Pauling figured out, and that is called beta sheets. So instead of this spiral staircase, we have these switchbacks. So it goes back and forth, kind of looks like a pleat or layers of sheets, right? So beta sheets. So we have alpha helices and we have beta sheets, okay? And these beta sheets, again, can take on more complex structures as opposed to just those long singular helices. We have these beta sheets here, which can form and bend and wrap around. And we can form different types of barrels. We call these beta barrels. So here in the middle is a receptor. So it can form a barrel around a specific molecule and maybe hide it or receive it and send it as a signal. And here on this side is a very large protein. This is called a transporter protein. So again, we have these beta sheets that wrap around and form a barrel, and it can sit on the membrane of a cell and basically form a channel or a tunnel for things to go in and out of the cell. These are all proteins that are genetically encoded by our DNA, but this is what they make. And they're the ones doing all the things inside of the cell. One of the most famous proteins that is made out of beta sheets is this protein here called GFP. It's the shorthand, but it's green fluorescent protein. And it was actually isolated from a jellyfish a long time ago, not that long ago, and it won the Nobel Prize because now with this protein, which actually fluoresces green, you can see here in this very cool microscopy image, we can put this inside of cells and we can visualize them under a microscope. And we can now also change this GFP and make it be all kinds of different colors. But it was first discovered in a jellyfish where it's naturally occurring. And what we can do, or what nature can do, is actually combine all of these structures into even more complex. So the ones I've showed you are basically all alpha helices or all beta sheets. But what we can do is make them even more complicated. So here, instead of actually showing the spiral, they're kind of represented as these rods. And in the middle, we have those beta sheets. This is called a Tim barrel. So this is a very common structure that we see in all kinds of proteins. So we can make them more and more complicated by adding more and more amino acids together. Here's a very small protein. It's an example of this. This is a toxin from a scorpion. And this actually will interact with our nerve cells and cause quite a bit of pain. I've never been stung by a scorpion, thankfully. But you can see we have a few of these beta sheets here. They're connected by some loops that don't really adopt that structure. And then we have this nice little alpha helix here. So this is not a super complicated protein, but it does showcase how we can combine these two different types of structures that Linus Pauling discovered. And what I particularly find fascinating about proteins is this idea of the structure of the protein itself can tell us a lot about what that protein does. Right? There's a lot of different structures that we could observe in nature, but there's a lot of them that are the same over and over and over again, and they do the same thing from one organism to the next, because that structure is very, very important for how that protein can function. And so now we're going to move away from this ribbon diagram for a second and add back in some of that detail just to show you why it's important to have that and how those little details can change the overall structure, right? Remember what I said? We're stripping away a lot of this chemical information when we're looking at a protein like this. And these two look very similar if we just look at this ribbon diagram, right? The top one is pardaxin, which is a toxin that fish make to repel sharks. And this one at the bottom is actually from the bubonic plague. So this is the toxin that makes. And you can see, again, they're very similar. They have these two alpha helical structures. But if we fill in some of this chemical information from the different amino acids, we can see that they start to look a little bit different. And their overall 3D shape looks a little bit different. And then if we even fill in that space a little bit more and understand where that chemistry is happening, we can see that they start to look, again, very different. And that's because these two toxins do very different things, and the structure for that is very important. So while visualizing our proteins like this can be very helpful, we can't forget that there's a lot of other information there that dictates how this protein will function. And one of the best examples of this is an antibody. So an antibody is a protein that we make, and it helps with our immune system. It recognizes foreign things that might come into our body, like a bacteria, and it interacts with those foreign proteins, detects them, and binds to them. So it interacts with them and then sends that signal to our body that this does not belong here. So over here we have this antibody, which again, these rods just kind of represent those staircase structures. And on this side, we've zoomed in to just a small section of that protein. So in the top here is this antibody. We can see some of those alpha helices, some of those loops, and it might be a little bit more difficult to see, but there's this dotted line here, which represents the end of the antibody. And down here is the smallpox viral protein. So this antibody nestles perfectly into this protein because of the shape that it's taking, because of how those amino acids are interacting. And that's very important for how that antibody is able to function. If it looked differently, it would not be able to interact and detect this particular protein, right? And so antibodies can change quite a bit to recognize a lot of different proteins. That's part of their function. And this is true for a lot of proteins, all proteins. If you change their structure, you change their function. And one example of this that we write about in the book is this tau protein. So my grandfather was diagnosed with Alzheimer's several years ago and passed away. And a lot of people probably can relate to having a loved one suffering from Alzheimer's or dementia. And so I wanted to write about that in the book because it's very impactful and we can understand it from a protein perspective. So in our brains, we have this protein called tau protein. And it's in there, it functions normally, and it looks something like this. It has those beta sheets. But then as you get older, for a lot of reasons we still don't necessarily understand, that protein unravels, loses that shape, and it refolds and takes on this new structure here with these alpha helices. And you might think those still look pretty similar, but this shape here completely begins to destroy in the brain. And they actually begin to clump together. So these proteins unfold, and they trigger more of them to unfold, and they cause these large clumps in the brain, which is what we believe is a factor in Alzheimer's disease. So again, this is just to show you that the structure is very important to how a protein functions. And a lot of the diseases that we see and observe that are linked to proteins are because something has gone wrong with the shape and the structure of that protein. I'm going to turn it over to Shahir now, and he's going to talk a little bit about how we actually can figure out what these proteins look like and why that's important moving forward with biochemistry.
I want to start by asking this question. Can we see proteins? So, Maggie has shown us a lot of really great structures and drawings of what proteins look like. The question is, how do scientists actually look at proteins? Well, we know that proteins are very, very small. They are tiny, in fact. And generally speaking, if we want to look at tiny things, what do we usually do? Well, we can use something like a microscope. We can use something like a microscope. The issue here is that proteins are very, very small. They're much smaller than microscopes can actually see. And here is a picture of Maggie with one of the first microscopes. It's very hard to see. One of the very first microscopes that were ever invented, a Leeuwenhoek's microscope with a very, very tiny lens. And that's when we started to look into the world of tiny, tiny things. And so if we look at the range of sizes, this is a logarithmic scale. So every time we go from one to the next, we're going by 10 times. And so, of course, we see things that we can see with the naked eye, like a chicken egg or an adult or a grown-up. And those, no problem. But then as we get into smaller and smaller structures, we would need something like a microscope, like a light microscope, for example. And we can see things like animal cells or plant cells, or even things that are smaller than an animal or a plant cell, like a bacteria or a mitochondria, or even some types of viruses. We can see those on a light microscope. But then proteins are much too small to see with a light microscope. They're in that range around 10 nanometers. And a nanometer is about a millionth of a meter, 1 millionth of a meter. And so what can we do? Well, first we can talk about how microscopes work in general. And if we have this sample of a fly, for example, we can shoot it with light. And what happens is that the light will be scattered, and we can use a system of lenses to refocus that light back and then recapitulate that image, but now it's going to be magnified. That fly wing is going to be much larger, and we're able to see details. And we can do this with a light microscope using the light in the visible range. And that's fine. And we can use that to see something like a fly wing or even some cells or even mitochondria or bacteria because of the wavelength of the light that we're using. So a light in the visible range, the Roy G. Biv, the rainbow that we see, have relatively large wavelengths. They're still very small, but they're relatively large, which means that we can only see things like a fly wing or a bacteria. But if we want to see something that's smaller, we have to use much smaller wavelength of light, much shorter wavelength of light. And that moves us into the realm of X-rays. And so we have to essentially shoot our proteins with X-rays. And there are large facilities that can do this. And essentially, it's kind of like a microscope, but it's definitely not like a microscope. First, we have to take our proteins and turn them into crystals, the way salt crystals form, which is a very difficult process in of itself. And then instead of shooting them with visible light, we shoot them with a much shorter wavelength X-ray beams. And then the same thing or a similar thing happens. The same thing that we see with scattering here, we see diffraction or scattering as the light hits the proteins that are in this crystal. But instead of a lens system, we don't actually have a material that can refocus X-rays the way that we do with a lens that would refocus visible light. And so all we're left with are the diffraction patterns, that the pattern of light that diffracts and scatters as it hits that sample. And we get these spots, and then it takes a lot of math and a lot of nerds over here and computers to actually figure out what this diffraction pattern can actually mean in terms of a real protein structure. So we're not really seeing it under a microscope the way that we would see a fly wing under a microscope, but the concept is very similar. shoot it with light, it just has to be the right wavelength. And again, why do we care about structure? Because as Maggie mentioned, many examples, protein structure dictates function. Here's another example of a protein that has these alpha helices that straddle this DNA, this double helix here. And by doing so, they can control when these genes are turned on and turned off. So again, very important. And then that brings up another question. How do proteins take the form in the first place? As Maggie mentioned, proteins are made of strings of amino acids, and they're kind of like beads on a string. This repeating pattern right here, and I've colored each amino acid differently here, and somehow they go from this sequence of amino acids, one of 20 different amino acids at each position, to these nicely folded structures, as we call them. So how do proteins fold into the structure? And this has been a mystery for about as long as we knew that proteins existed in the first place, until very, very recently, around 2020 or so. These two individuals, Demis Hassabis and John Jumper, working at Google DeepMind, developed a tool called AlphaFold. How many of you have heard of AlphaFold before? Yes, excellent. AlphaFold has really solved a lot of these issues of taking a protein that is a sequence of what we might call beads on a string and how they actually form into these precise three-dimensional structures that allow them to actually function. And this was accomplished by the help of AI, artificial intelligence, essentially by using these neural networks. And this is a network of essentially computer neurons, in a sense. And just like we can learn things by experiencing them, neural network can do a similar thing. So all you have to do with a 4-year-old or a 6-year-old is show them pictures of a few dogs, and then anytime they see a dog, they'll probably recognize that it's a dog, even if it's a different breed or a different color. And it's a similar thing that we can do with neural networks. We can feed them some of the known structures, about 200,000 or so known structures of proteins, and they can figure out the patterns of how to take a sequence, which is represented by the letters up here, a sequence of amino acids, and then tell us how they would fold into that three-dimensional structure. And they've done this with such great precision that they earned the Nobel Prize in Chemistry in 2024. But it turns out that was just a half of it. In fact, they shared this prize with this individual here, and that's David Baker at the University of Washington. And very simply put, his contribution was the opposite. Instead of going from sequence to structure, can we come up with a structure that we want to design? We want to design this structure. Can we figure out what sequence will give us that structure? And so it was kind of working at the problem from the other end. And this really opens up this field of protein engineering, where if we can get new folds and new structures, we can get new function. And I could really talk more and more about protein engineering because it's near and dear to my heart, but I'm going to leave you with just a couple of examples here. So in 2017, Science Magazine published this saying in the next 30 years, we'll make 4 times more plastic waste than we ever had. And being good ancestors, as was said earlier, is really about taking care of the trash heap that we leave here. And this is something I'm very excited about. This is one example of a protein that was engineered, an enzyme, in fact, that can break down plastic. And there are more and more of these examples of enzymes that can break down plastics so that these plastics are no longer non-biodegradable. They can actually be biodegradable. And in fact, they can be broken down and not left for future generations. I want to talk about just a couple more examples here. In terms of carbon capture, we could teach enzymes how to be better at taking carbon dioxide from the air. And an example that comes from our own lab at Indiana University South Bend, where we took a bacterial protein and trained it to bind to the active ingredient in Roundup. It's known as glyphosate. It's the most commonly used herbicide in the world, certainly in the country, and a suspected carcinogen. And when this protein binds to or grabs onto glyphosate or the active ingredient in Roundup, it sends a little fluorescent signal, it sends a little bit of light that we can detect. And some of these examples of engineered proteins you may have also heard of. Biologics like Avastin or Herceptin all of those are engineered antibodies that can be used as anti-cancer agents, very, very successful, and something like Humira, for example, for inflammation, autoimmune. And so, just to close, I just want to talk about the book a little bit and say a couple of things. Yes, there's a lot of information in the book, but the book is really a book of stories. We talk about stories of some of the scientists that we mentioned here today, but we also talk about our own personal stories, growing up in different parts of the world, and some of the really important things about us, but also how it relates to proteins. This is really about communicating science through art and storytelling. We live in a very divided world, as I'm sure you know. And it's very easy to get into a yelling match with someone. There's a lot of distrust in science, and we hope to make a difference in that just by changing the conversation a little bit and by fostering fascination. It's about going back to that place where we were fascinated with proteins and their structures when we first learned about them in high school or college, and to share that joy with people. And this is just part of some of the efforts that we do in terms of science communication. The book is one of them, but we also have been trying to train other scientists to be, better science communicators, especially with when facing conspiracy theories. And also, we really are keen on using art to elucidate some ideas in science. And these are some of the pieces that we worked on for a science communication conference, some of the work that we've done to just kind of elucidate some of the things that are in the book. Of course, you have the iconic alpha helix right here. And again, I can talk more and more about proteins, but I think I'd like to end it here and thank everybody who invited us to come here. Thank you very much.
Well, thank you. That was wonderful. Thank you for celebrating proteins over DNA. Um, some of my colleagues often are upset that everyone is so dismissive of proteins when you clearly illustrated that they are the key to how cells work. So I'm just going to start the conversation with a few questions, but I'm sure you all have a lot. So I'm going to try to be cognizant of the time. So just some things that are on my mind, and I love proteins. One thing that's I've been thinking about a lot, as have others, is one of the big events in synthetic biology before AlphaFold, which is the field I work in, was the creation of a so-called minimal cell by the Venture Institute. This was done almost over 10 years ago, I think. And so this is a cell that was synthesized, which has the smallest number of genes for life. Pretty remarkable. But what's curious about that is that they still don't know. They scratch away at it, but I believe they still don't know what 25% of the proteins do. And they are very similar to proteins that are found throughout nature. So I wonder what your thoughts as protein experts and people who think about how we're going to figure out proteins, what do you think about that? How are we going to get there? And do you think we'll ever completely understand a single cell?
So full disclosure, I am actually a microbiologist by training. So I did some biochemistry, protein engineering work. So this idea of not knowing what genes do is very near and dear to me as a microbiologist because we have no idea what bacteria are doing most of the time, and there's so many proteins that we can't identify. We know they're there, but we have no idea what they do. And I think two things. Maybe this is a bit of a cop-out question. I think it's just a lack of imagination sometimes on the part of scientists, that we're really pigeonholed into this must be what we should be the question we should be asking. And Linus Pauling is a huge inspiration. Like, what if we just played around a little bit and had a bigger imagination about what could possibly be going on? And I think there's a lot of cases too where we see proteins that are moonlighting and doing more than one job. So we say, okay, we figured out it did this, and we miss, and we move on to the next thing, and we don't realize that proteins can be doing more than one thing at a time. And it's easy to kind of get that one paper out, say we figured out it did this, and then we kind of forget about it. And so I think it could be both of those things are happening at the same time. And I don't know if Shahir, if you have any thoughts about that. I don't know if that completely answers it, but I think, yeah, that would be what I have to say about that.
No, this was perfect.
So when I began my life as a molecular biologist, or before that, the cell was often referred to as just a bag of enzymes. And we now, thanks to elegant microscopy and molecular biology, now know that even in the simplest bacterial cell, there's a huge amount of organization. Now that we can start to tease apart all these proteins, and in particular enzymes, one thing I wonder is, and this goes to what we call the green economy, do you think that we'll be able to replace chemistry with biology?
Oh boy, can we replace? So, well, so I've worn many hats over the years, and I actually work in a department of chemistry and biochemistry. So, the basis of proteins' work is chemistry. So, proteins are nature's brilliant chemists. You can put it that way. Again, this might be a cop-out. Can we replace chemistry? Look, here's the thing. In the 1950s, maybe earlier, there was this idea of solving the world's problems through chemistry. And later on into the 60s and 70s, and we get things like Roundup, for example, which was put forth as the safe alternative for previous pesticides and herbicides. And in fact, it was, in many ways, a much safer alternative. And chemistry just kind of kept getting better and better and better. But there is a limit to what you can do with chemistry. So if I have a headache, I can take some Tylenol. That's chemistry. I don't need to take an engineered protein to help with my headache. But when we're dealing with something like a cancer or an autoimmune disease, we can treat it in both ways. And so far, or at least for now, a far more superior approach has been the protein approach, the protein-based approach. And there's a reason for that. We see that proteins are far more complex than a molecule like Tylenol or Advil. Again, those have their place, but proteins can be far more specific to target something like a cancer or an autoimmune disease without having these off-target effects that something else might have, like a chemotherapeutic. That's at least from the therapeutic point of view.
Thank you. That was a great answer. Very inspirational. I happen to work on protein therapeutics.
Yes.
Thank you. Then I also, so you mentioned AlphaFold, which of course is just an amazing leap forward in principle. But why do you think, it's interesting that that was actually not developed by chemists or biologists. And so why do you think that is? And, were the biochemists just being slackers or what? and maybe, I don't know if it's a correlate, I also want to get this in before we go on. And I think you can intertwine the two. But we are also on that synthetic cell front moving towards an expanded genetic code where AlphaFold and all that, all those predictions are going to go into a whole new direction. So can you take all that and unpack it?
I have something to say about the first question about were biochemists just being slackers, um, which, could be true. I, it's been the protein folding problem has been a massive problem for a very long time, and everyone has been trying to work on it. And I'm not an AI expert by any means, but we've spoken to several, and AI and this neural network learning is perfect for solving a problem like protein folding. There's a lot of things AI should not be involved in at all. This one is perfect for solving this particular problem. I'm sure there'll be things that we continue to learn and need to adjust, but the way that this type of AI it works from, again, I'm not an AI expert, but um, this problem was the perfect way to apply it. And I think, oh, so go ahead.
I had one thing, maybe you're going to say this, that it was also perfect because of the work that had become
Correct.
before.
Yes.
Maybe, why don't you talk about the protein database?
Yes, so if you remember from the slides that the AI, the neural network had to learn from something, the way that children learn by looking at pictures of dogs or petting dogs, there had to be a foundation of what proteins actually look like. So it didn't come up with proteins on its own. It had to learn what proteins look like. And there is an entire data bank called the Protein Data Bank. We're very creative in the way that we name things. It's called the Protein Data Bank, and it's just an immense project that really collects all the structural information that we know, all the 3D information about every single protein that has been determined. And it's available for all of you, by the way. That anybody can access any protein that's deposited in there by going to the Protein Data Bank. And so there's a lot of work that's been done. And I think that to your question about, why are we slackers? I think this really celebrates that diversity and importance of diversity in science, that we can say, oh, we missed this one, and somebody else did it. But it's really about bringing in people from different points of views. And I want to point out also another person that we talked about. That was Jane Richardson. She's the one that came up with the ribbon diagram. She was one of my professors at Duke University. And she was never trained as a scientist to begin with. She came from the humanities, and she worked with her husband, Dave, who was a crystallographer, someone who would shoot crystals with X-rays, right, and determine structures. And these structures are just so cumbersome, very difficult to look at. And she came up with this idea. And again, it's about opening the doors for people who come from very different backgrounds to try to help us see a problem from a different point of view. And I think that this is, can I talk about my mantra here for a second? I feel like scientists, we close the door on a lot of creative people because science has gotten a bad rap, and it's been the myth is, and this is not true at all. The myth is that creative people go into the arts and the humanities, and the non-creative people go to the scientists, which is absolutely wrong, absolutely wrong. And I don't know who goes around and advertises this, but I've seen it with children. They say, well, I'm creative, I love art, so I can't be a scientist. And this is very prohibitive, and it's a problem that we really have to address because we see what happens when creativity comes into everything that's been invented has been thought of before it was invented by a creative person. So we really have to have that diversity and creativity in science. That's my mantra.
Thank you for those words. I fully agree with you. Um, I think now is a good time to ask you all for your thoughts and start with you. And I believe there's a microphone. So my childhood hobby is origami, and my question to you, and it was not trivial, at least at that time, to explain how things fold. And I wonder if you could maybe give us a taste of how do you describe the folding structures of proteins? I don't expect you to kind of give it to us completely, but give us a taste of what kind of vocabulary, what kind of methods you use to actually describe proteins.
Yeah, I can jump in, and the answer is it depends on who you're talking to, because being a good communicator, someone who is going to communicate something, it really depends. So the idea here, very simply, is that it really has to do about attractions and repulsions. So there are certain parts of the protein that are going to attract each other, so they're going to fold towards each other. And there are certain parts that are going to repel each other. And it's that interplay between attractions and repulsions that are going to form the final three-dimensional structure. But there's also a reason why we see these patterns repeating. There are, again, hundreds of thousands of protein structures out there, but we always come back to those two basic forms, the alpha helix and the beta sheets. And as you might notice, they are repeating structures. A circle or a spiral is a repeating structure because it's the repeating nature of amino acids as they go on a string. And there's attractions that naturally happen. We call those hydrogen bonds. And because of the way that the geometry works, those are going to be the two more common ways of actually making things work without having things clash into each other, repel each other, and having also that balance of attractions. It's about attraction and repulsion. And what we hope to do in the book is use these illustrations to show the three-dimensionality of, of these structures. And what's really cool also is you can go to the Protein Data Bank, right, rcsb.org, and you can look up protein structures. You can turn them around in 3D using your mouse pad. That, I think, is the most powerful way to describe how a protein folds, is by looking at the three-dimensional structure rotating around.
Well, you can also go to AlphaFold yourself.
Yes.
And fold a protein.
Right. Type in a sequence and you get a protein. Yes.
This may be a related question, but we were talking about fit, things fit together, the enzymes and viruses. And when I think about fit in macroscopic world, it's very mechanical. It's gravity, it's structures, and elasticity. Fit in the microscopic world has to be something else. And with attraction, repulsion. What's the physics? How does this analogous to geometry of the macroscopic world?
Excellent question, because things like gravity, for example, are not really going to play a great role. But because these amino acids that the 20 amino acids are kind of like colors in a crayon box, and you can just use, each one has its own property. And what we know about attraction is that opposites attract, right? So some amino acids have a positive charge and some have a negative charge, and we know that opposites attract. I always tell my students, remember that opposite attracts. This is why I have terrible friends. So this is no, they're wonderful. They're absolutely wonderful people. So opposites, so you can have a positively charged amino acid over here, a negatively charged amino acid over there, and they will find each other and attract, and that will cause a folding. There's also another very important thing that I think people may not think about, and that's water. Proteins in our bodies are swimming in essentially salt water, and there are some amino acids that are water-loving. We call those hydrophilic, and there are some that are water-fearing. We call them hydrophobic. And if you have a string of these hydrophobic, water-fearing amino acids, they want to hide away from water, and they will drive themselves towards the inside, the interior of this three-dimensional structure as they expel water away from themselves. That's a big driving force for folding as well. So those are generally what you have. Charges and then interaction with water.
When we were talking about the enzyme and the proteins that were engineered to develop plastic, how do you get replicated?
Yeah. I can take this one too.
Well, Shahir is the protein engineer by training, so I'm letting him handle some of these questions.
So as it turns out, some of these enzymes were actually isolated from bacteria that were found in dump sites. And bacteria are basically evolving right in front of our eyes. They do amazing, amazing things. And Maggie can tell you all about bacteria. But they were isolating some enzymes that can do just a little bit of these reactions. They were very slow, but they could digest it. It's not something that could be done on a massive scale. And then what protein engineers do is they start from that point. That's a nice starting point to have, what nature gave us. And then they start to make mutations, which are essentially changes in the individual amino acids. And you can focus, if you know the structure, then you can focus on the amino acids that are close to where the plastic binds before it gets broken down and make some changes. And you can use computer-guided changes, or you can just look at the structure and make predictions based on what you know about the laws of physics. So there are many different ways to do it. You can also do something called directed evolution, where you can make random mutations and do a type of selection. You select for the ones that are functioning best. The way that nature does it, but you do it in a test tube. So there are many, many ways to improve something that already exists. But it started with bacteria. Bacteria are everything. Yeah.
So looking ahead at the development of tools like AlphaFold, who do you see as the most innovative users of these new tools in the synthetic biology or academic research, labs or private sector companies that are applying these tools in the field?
Oh, I don't know. I don't know.
Yeah, I'm really not sure. That's not a field that I'm involved in and follow very closely. I'm, like I said, a microbiologist, so do pretty basic microbiology and infectious disease. So um, I don't have an answer to that question. Shahir, do you track
I
any of that?
think I think Pam might
Yeah, I
have the
think
best answer
yeah,
to
she
this.
should
You
answer
know,
that.
Sure.
yeah.
At the risk of, yeah, I don't want to.
No, please you share with us. You're the expert
I think
here.
we're at a tipping point, actually. Synthetic biology, which is this idea that we're going to use biology like chemistry to make everything. Um, we 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 kind of proposed that about 20 years ago, and we haven't made everything.
Yeah.
And so I think we're at a tipping. I hope we're at a tipping point now where some of these new approaches and tools that are happening will actually kind of reinvigorate the industry, and you are starting to see that happen.
Hi, I'd like to ask a question from the science communication perspective. I'm curious sort of what drove you both to write this book and if there was sort of a gap in communication about proteins that you're looking to fill.
I think there absolutely was a gap in proteins that we wanted to fill. I mean, there really wasn't anything that was accessible to people who've never taken a biochemistry class, right? By the time you take a biochemistry class, you're probably a senior in an undergraduate program. So even for people who went to high school, they might not have ever encountered that. So there's stuff out there that's a little bit more technical, but to have something that's accessible, especially in light of how AlphaFold was Nobel Prize last year. This is something very recent, and people do not have the tools to understand what's happening in this field. So we started writing it about 5 years ago. We had the idea, and I'll let Shahir talk about that briefly too. But we both just loved proteins and felt that that was what we wanted to write about. And there was a massive gap, right? Trying to explain to my friends and family who never went to college what I was working on was very difficult, and we wanted to give that information to the people in our lives. And we wanted to do it in an accessible way that wasn't scary, right? It's already scary enough to read about DNA. That's something that people are familiar with, but still there's barriers. So then taking the leap to something that they have really no idea about was very important to us. And we were already kind of thinking of ways to communicate to people. And the art part was a huge component. I was actually taking a creative writing class when the art part came to us. I started drawing protein structures. It was a graphic memoir style class, and drew these protein structures, presented them to my non-scientist classmates, and realized that the illustrated component was a huge part of being able to communicate proteins and protein structure and function to people, because like you hopefully have appreciated how diverse and beautiful they are. And so that was really, at least for me, and I think Shahir as well, the driving force of we have to talk to people we know every day about what we do, and it's very difficult. How can we make this more accessible and fill this huge gap in the popular science world, at least?
Yeah, just a tiny bit to add on that in terms of just science communication in general, that we're both very passionate about communicating science beyond this book as well. I mentioned a few things like workshops that we give to scientists on how to be better communicators, but also in the face of misinformation and things like conspiracy theories. We've met several flat earthers, wonderful people, by the way, and it's nice to be able to engage with people without yelling at each other, because otherwise that just really increases the gap in understanding. We talk about just some general techniques on how to handle these types of conversations and to try to really make an impact instead of just creating impasses. Right? So that's one of the reasons why we wanted to write this book. It's just part of the catalog, I guess.
All right, I believe we have time for two more questions. Hello. I'm intrigued by the title. I'm hoping you can address whether North is a metaphor or if it's, I hope so, it could be. So,
Oh, great question. I guess we never really explained what you have to buy the book. No, I'm just kidding. Uh, we encourage you to. It's so the as we wrote this book, even though we knew a lot about proteins, we were still discovering what some of these proteins do, not just within our bodies, but within the bodies of all these different plants and animals and so on. And we came across a class of proteins known as cryptochromes. And these are all the way from bacteria to humans to everything in between. In plants, they help the plants reach to the light, and they grow in different seasons and figure out what day-night cycle. For us, they help us with things like jet lag and resetting our circadian cycle. So they're light receptors. They just help us understand light. And in some migratory birds, they have a very, very unique property. They actually, using light, they can use the energy from the light to make the world's tiniest compass out of a single electron. And electrons are actually magnetic, so they can detect the magnetic uh, pole of the Earth. And based on that, it actually sends as far as we know, sends a signal through the optic nerve of the bird. And as far as we know, the bird perceives a color that to it means north or south. And as it turns around, it can actually see it through its eyes. I know it's insane. It's really crazy. And so this is a color that I don't think we can ever perceive, but yet we can learn about it. And that, to us, I thought was very exciting. And that's where the color of north comes from. So it's not too metaphorical.
Uh, I was wondering, in terms of functional engineering, if we incorporate non-natural amino acids in the future, would we be able to access structures not possible, or is 20 enough?
Excellent question. It turns out even in nature, 20 is not enough. The reason why we take things like vitamins is because they add extra chemical diversity to what we have. So you can make a protein out of the 20 amino acids by mixing them, at every position. You can pick one of this. It's like coloring again. You have the box of 20 crayons, but sometimes you just need that one blue that is not there, and you have to get that from something else. So we already kind of use non-natural, they're not amino acids, but we already use extra chemistry. So then the question is, can we incorporate non-natural amino acids? And that's already been done, actually. So excellent question. That's already been done. There are people who are doing this. And you get new chemistry because you're incorporating new chemical entities. So yes and yes. Yeah.
Okay. Well, thank you so much. Thank you for writing about my favorite thing. And I just say, buy the book and love your proteins.
Love your proteins. Yes.
Thank you.
Thank you.